All right, you guys. How are you doing? You did good job. I'm working on the test. Looking good. How are you guys doing with all of that? How are you feeling about that? Well, we only have four people, just for share or don't share. It's all right. um, I hope we're gonna get a few more because I want to go through sort of the pathology project stuff, but that's okay if we don't. um, so yeah, that's what I wanted to do today. Good morning, everyone. It's a little early here for starting. It's well 10:03. We can get into the week if we want. So now that we finished the muscles and the bones, they are behind us. You can clear your mind from all that weird terminology stuff. And we're gonna go now. We already have gone really forward into the good morning, into the into the heart, and now and then the blood, and then slowly we're working our way to the brain. We have a few chapters which have no coloring labeling. We have many chapters that do. They're much less extensive than the bones. The muscles, particularly with all the writing, they're really just labeling. The organs are a little bit, we're doing a little bit less anatomy and a little bit more physiology on those kind of things. And so as we go through this material, it'll be crucial to make sure you go. Oh, and did I open these weeks? Jesus Christ, I'm back again with my problems here. Make sure I have open all the weeks. But make sure on this material you work very strongly with the test review that's in the test three last week material or after the last week's material. So go all the way forward to that review and look at what I want to for the test material. Let me actually just jump there real quick since I'm just zooming there. And then make sure, where would that be? Let's see. Safari. All right.
Yeah, you have until
All
week 12 on this. I don't know if you're looking on the student view, but the
yeah,
student
here we
view,
go. I got the week 12.
yeah,
So that's good. That's great. And I'm going to finish the rest today. And then under here, we go to test three under the review. Um, I want to make sure from the start you're going with this test review document. Print it out if you can. Otherwise, somehow put it on a device or something. Because see, as we go through the heart last week's, this is what I want you to know from the heart lecture as it comes to not the terms, but the function part, the physiology part. So it's not that many bullet points. Some chapters, of course, are more than others because blood, we don't have much as an anatomy stuff there. It's much more physiology. What do the different blood cells do and so forth. But for the most part, the whole rest of the chapters in terms of the physiology portion is now condensed to these 5 pages at the end of it at the review. So the more you focus in and make sure all of those bullet points you understand, and of course the other stuff too, but that's the stuff that's going to be on the test for sure. And so make sure you get those and reach out if you don't, or use those to make flashcards or so if you want, or use your own words. But um, and if some of this stuff doesn't make sense, make sure I still have to go through it again. It's been a while. But it should be all fairly sensible the way I might use RBC instead of red blood cells or so. But once you work through the chapter, you know what that means. So make sure you be aware of that as you work through the material, because that way you can relax. Well, whatever that means when you study and do school, but relax as much as possible into the material as you go through it, and don't have to always worry that, oh, this is on the test, this is on the test. Oh, the other thing, of course, that is on the test. Let me just finish. Well, sorry, I should have stayed in there. Mentioned that too.
when is test three? Have you
The end of
told
the
us?
chat at the end of the semester, the last week, the test week.
Okay, the final.
Yeah,
Okay.
so this is a long period. That's why I want to structure it so much that you guys don't have to be worried from now next 7 weeks, for because it's a long period. But we also have the pathology project, and so I rather structure it that way that you can focus on watching videos, doing the questionnaires, and then make sure those bullet points you understand. And as we get closer to the testing, you can go back and review those, and then you'll be okay for the test. Does that make sense?
Yes, thank you.
And then the other thing besides the review part is the anatomy term list, and that one is the same as we have for test 2. There's just a list with a lot of terms, and we're going to go through those terms. This is just an accumulation of the terms that we go through each chapter. So that's also a good one to print out, because then you kind of, as you go through it, make sure you get those, and, can start the ones that are harder and go through them a few times and go back at them and stuff like that. And so for the test, the last test, you'll be ready for that. Because the last test is a combination between test 1 and test 2. And so it's got some multiple choice true false, and it's got some fill-in stuff in it. And then I also have review videos here. That's for later as you get closer to the test, I think, when you go through it, and you just watch those a few times if you'd like. Not everybody needs to take advantage of all these different tools. I'm just decided to share all the stuff that I'm doing or that I have done, or as much as that makes sense, because we have all different learning styles. So, if it's a little, this is so much material, you don't have to look at all the stuff. It's not mandatory. I also made a picture document here that might be good to download versus maybe print out. Is a little too much ink, depending on what kind of printer you have or accessibility you have. But that's just I'm not going to download it here because it takes a while. That's just a document of all the pictures that I use to label then for the exam, for the models. All the models are there. So that's pretty much that as a starting point. Now I wanted to let's go back and see if you guys have any questions, any concerns, any comments so far, how the test was. We have a couple more people that added on. And then we go back into this week, which a lot of this is going to be pathology project this week. How you guys doing? Did you guys? I'm sorry, there was a few people. You guys had some technical challenges with the test. So if that was you, and you felt like, oh my God, I was frazzled, I couldn't focus, and that got so bad because of that or something, make sure you reach out. We can talk about that. And I
Yeah,
don't.
I didn't know that it was timed actually, the test. I just realized it when
Yeah, for
30
the
minutes
three
was
hour.
left.
Oh, you just ran out. Well, we can
Yeah.
talk offline. Yeah, we can figure that out. I'm going through it, and I'll catch those things and I'll reach out. I'm not gonna be a hard one. This in some ways is also a basic an intro class. Not every teacher in
Thank
a
you.
higher class is going to be doing that, so be careful about
All
that.
right. Thank you. Appreciate you.
But yeah, no, I mean, this is about you guys multiple ways repeating the information, and then one of those is the exam. And so it's not going to be, I'm not going to hang you on that. I know how there's a few that don't participate much, but they probably don't even hear this video. So that's always the case. It's hard to do online class. It's I myself too. Sometimes it doesn't fit in and it just glides away. You just have to do it again. That's all right. We're not punishing on that. But so if that's okay, and again, I'm going through the tests today, all of them. Make sure have a little patience with me on that. Hopefully, also the grading challenge. I'm sorry about that last week that came up. Hopefully that'll be resolved after I go through grading today, and everybody's back on track. Otherwise, reach out to me. I don't want you to have anxiety about that. Please, lay it on me on that one. Anyway, with the grading the canvas, it's always at the end. It's because it's hard for me sometimes to be on top with the Padlets or so, and so the grade might be fluctuating. And at the end, everything will be fine. You can assure yourself, if you do all the work and you don't try to slide, you're going to be fine and get an A in this course. That's the way it goes. If you are not, then we'll figure it out and talk about it. If you really put in the muscle, the brain muscle, you'll be fine. So let's just go through today's material for a moment. We're gonna cover. Wait, we did the heart, right? What are we gonna cover this week? We're gonna cover blood. Blood, right? Shoot, did I pull up the wrong week? Yeah, we're in week 10. So we're going to do blood and we're going to immune system. I pulled up the wrong weeks. So hold on, let me pull up this stuff real quick. Oh, that's some big stuff. I like that stuff. While I'm pulling that off, I also liked I'm starting to look into the pathology topics. They're looking good too. I'm happy with what we got. And I started thinking, depending on who you are in a group and how versed you are, maybe this time some of you, we don't want to do it in a PowerPoint. Maybe we'll do a brochure or something like that. I can help you with that. Figure out, make it useful, our work. All right, let's go a little bit through the material, if that's okay. And again, if you have different thoughts or want to not listen to this stuff or it's too boring, let me know. We can move on to other stuff. Just interrupt me. Also, if you have questions, just interrupt me. But this week's material is, for the most part, physiology material. We have that little lab thing where we label the blood cells, but that's really small, not a big deal. So we're going to go through blood. Now, we did the heart that pumps the blood. We did the blood vessels that moves the blood around in the body or channels it through the body and perfuses the tissue. Now we're going to look on the inside and what's that blood thing all about on the inside. And so we're going to have blood is a connective tissue type. So it's a fluid connective tissue. It's a transportation tissue, if you want to call it a tissue. It's kind of fluid, but it's still a tissue in terms of our analysis of talking about these things. It's got volume to it. And when we look at connective tissue, we always have a cellular component and an extracellular matrix component. And that cellular component is the ones we all know is the red blood cells. That's why the thing is red, because of the iron in it. And then we also have white blood cells, and those are helping with the immune system. And then we got platelets. And the extracellular matrix is blood plasma is sort of in bone, that's the calcium, that's the hard stuff, that's sort of the outside the cell stuff. And then we always have a fiber component, and in the bone that was a lot of collagen fibers that holds that bone sturdy. And that's, bone is sort of the hard connective tissue. That's sort of the opposing one to the fluid one, which is like, really, they're both the same thing? Yeah, because they have the same components. And then in terms of the fibers in the blood, we have fibrinogen, which when you see a word with a GEN at the end, that's from a precursor. In the the easiest example to remember is Genesis in the Bible, that the creation. So this is before we're finished, fibrinogen. And so when we change fibrinogen, it becomes fibrin. And when we have fibrin in the blood, we actually have blood clotting. And so for the most part, we don't want that thing to be clotting. We want that thing to be fluid. And so we don't have it in a real finished form that is functioning, but we have it in a precursor form, chemically speaking, that's not quite finished, and therefore it is not functioning. It's like you need a couple more keys into this molecule, that fibrinogen molecule, and then it becomes a real fiber, and that fiber then is called fibrin. This is a very complicated process to change that, because as you can imagine, if you have a blood vessel that needs fiber, it's used in the blood to patch breaks in the blood vessel, and then you need a patch material for that, and that's the fibrin. But and then the blood component platelets, which are little particles that float around in the blood. But if we don't want that break to be patched, we don't want that clumping to happen because that's going to give us a stroke. And that will clog a blood vessel, which is what a stroke is. And so there's some interesting stuff in here. So these are the main foundation components. And then, the chapter goes through how much blood we have, etc., where it's made. It's made in the bone marrow, which is interesting. And then we go into the different functions, and we use it to transport oxygen. We kind of know that part, I think, for the most part. Oxygen goes on the red blood cell, which is the main, the one that gives you the color. It's the most numerous blood cell that we have. And then we also have the white blood cell we talked about already, that defense, that's immunity. That's the next chapter. And then other stuff that goes through the blood is nutrients and metabolic waste and so forth. Hormones go through the blood. So it's a good transportation system. I'm thinking like, it's the freeway system in the body that goes through town and can transport stuff around. So that's pretty good. It also has to do with what we just talked about with that fiber and that coagulation, that's what that's called. When we have a break in a vessel, we need to close that. That's that process. And then blood is very has a lot of heat to it. So liquid water has a high heat capacity. We learned that early on in the chemistry. So it can carry heat around in the body, and that's nice because it balances things out. That you see that on the ocean, and when you're living by a body of water, you got much more temperature changes in the cold and heat and warmth or day and night than in places where it's dry, like in the desert, for example. And so it's the same in the body. The heat is carried around in the liquid, and water, and the liquid is the blood. So if you need to radiate heat off because the inside gets really hot and warm because you're, running the heat, the blood will go to the surface of the body and will radiate off and give you more, let go of the heat and give you more cooling for the body that way. The other thing is the sweating. We can carry the liquid and the heat out that way because, again, liquid carries a lot of the temperature with it. So it's interesting, that stuff a little bit. And then we go a little bit further into the RBCs. You guys can do that in the videos. The red blood cells, how many and what exactly they're doing and how long they're living. And they're not living long. They live 120 days. So they're making a lot of those. Look at that, 116 million each minute. Let me make this a little smaller. Oops. So that's pretty cool, pretty intense. And then from there, we move into the white blood cells. And when we look at white blood cells, we're looking at immune system. So now we're looking at some immune system. When you look at the immune system, you're looking at the body's defense. So when we live in a world, we're living in a world that has a lot of stuff coming at us, and the body needs to be able to defend against these things or keep the inside clean and make sure, we don't get bacteria or other things coming in and taking us as a host. And that's what the white blood cells are here for. And so we got multiples, 5 of them, different types, that each of them does a different function. And what we're going to do here, we're going to sort of connect the 1 or 2 functions to them each, and that's sort of a starting point for us. The other thing that you often do in an anatomy class, you're looking at identifying them, and that's the activity that we do when we identify them on a picture, basically. And when we look at that, we look at the different appearances, and that's where we group them into granulocytes and agranulocytes. We have 5, and so 3 of them have little dots on the slides when you look through the microscope in the background. And that all has to do with staining, the way that they try to make it so it's visible as a cell. So it's not really natural in that respect. But, when you look at the naming, it's kind of funny. It's like you see the granules here. You see some of them look a little bit pinkish or, a little bit like the background, and some look more reddish and some look more bluish. And the reddish ones are called the eosinophils. And so I can, the only way I could remember that when I first studied it was looks like neon light a little bit, although it's not yellow, but it looks a little shinier. And then these look a little bit more like neutral to the background compared to the other ones, so neutrophils. And then basophils, I don't know, basic, I don't know, for some reason as a base and acid, the base is a little more bluish. I don't know, whatever has. That's just my own brain. But so that's sort of, why they name these things. I mean, it's kind of silly in some ways, but it's good to know. And then the A granulocytes, there's two of them. There's just the ones we don't have these dots in the background. They have, A means they don't, without. A means without. And so we got two. We got one of them are monocytes and one of them are lymphocytes. And the lymphocytes, you guys know, the lymphocytes are the B cells and the T cells. We know them. They just start out as lymphocytes and then they become T cells or B cells. And then the monocytes are interesting too. They're big and they go to tissues and then they're just into tissues. They're not in the bloodstream and they're I mean, some of them are, but a lot of them sit in the tissues and they sort of clean the lungs and they become dust cells and they help the liver to stay okay. And okay, yeah, Andrea, hold on. And they were keeping the liver clean down there, and they call them Kupfer cells because they look a little coppery down there. So that's kind of interesting. Yeah, Andrea.
Sorry, I had a question. So
Yeah.
what you're going over now is what was due last week? I'm confused. Sorry, I lost where we
what?
are.
Oh, wait, am I completely confused? I've been having a hard time having straight because I'm at all these, when I make the videos, I get overwhelmed completely. But wait a minute. I thought this was,
sure.
I thought this week.
Week 9.
Help me out.
Week 9 was the heart and blood vessels and lymphatics was week 9. And that was that last week?
That was due last week, right? Or Sunday. Right. That was due yesterday.
Okay, yeah. So now coming up.
And so,
I think because week 10, it says here week 10, blood, but I think in the module, but I think that should say blood vessels because we handed in, we went everything up to, I mean, am I wrong for the other people who are on the call?
Yeah, please help us out because I'm looking, today's the 23rd, so I'm looking, the blood questions are due next Sunday.
Yeah, because you review what we're doing in the future, right?
Right, and I just like to do a little overview, yes.
Right.
And if I'm going to talk into the detail, let me actually know because we don't have to. We can do, that's okay. I don't want to repeat myself too many times or I don't have to. I think that's totally cool.
What you were just going over, the blood, that was last week. And this week we're starting with blood vessels, number 16, right? Blood vessels and number 17. I mean, I'm looking at, your little book.
And you're looking at, you know what? And you probably are right. I mean,
unless I went ahead because everything you were just going over, I did last week.
I don't have a booklet on me. But that's, I think, one of the things where I should have, and I change it because the booklet, first I go to blood vessels and then blood.
Yeah, you skipped the blood and went to blood vessel. So it was heart and blood.
I did. Yeah, I actually did because that's one of the revisions I need to make in the booklets because when I set up the weeks, if I do heart and blood together, it's too much.
Yeah.
The heart and the blood vessel go better together than the blood and the immune system go better together.
Okay, so week 10 is blood and immune system.
So the way that the class is built in the structure, as the classes last week we did the heart and the blood vessel. Did you guys? None of you guys do it that way? Because I can reverse it. It's my fault, really, at the end of the day.
I did the heart, and I just went to the book because I didn't go to the lab. So I did the heart.
It's all right.
And then blood. So the heart you labeled in the booklet 14. So 14 was the heart. And then 15 was the blood. That's what came next.
Yeah, I need to.
And now in the book, 16 is blood vessel and 17 is immune system.
Yeah, I just did the heart. I was looking at the assignments
Okay.
that was due.
Because we did the coloring of the blood, those leukocytes. That's why I was like, when you were going over this.
Oh, I didn't do that.
No,
Oh, yeah.
because it's due this week. So I think some
Oh,
of you guys did it through the canvas, and then some of
Yes.
you guys kept the booklet thing. And
Oh.
it's my bad because the last 3 years I've been building up the canvas, and now I got to go back and do a booklet revision. But everything takes about a year. It's so much.
Oh, okay. Yeah,
okay.
I just do
I'm sorry
whatever
about
was
that,
on
but
canvas.
yeah, no, and that's normally how it works. This is sort of generally we
Okay.
actually this is the first time I ran into this with the situation
Yeah.
right now. But let's pick it up. Where are you, Andrea, with where you at with that?
So yeah, so I did the heart and the blood. So this week I was gonna do blood vessels and the immune system.
So you're gonna be fine because blood vessel is I because the model in the lab was I didn't like it.
Uh-huh.
Once they had it was ambiguous about certain things a little bit, and so I ended up not really doing too many blood vessels as a
Okay.
test goes in anatomy, because also and there's not many on the list as you can tell, or there's none on the list, and the other reason is this: if you actually look at the name of the blood vessel, most of them are named by the organ they feed or by the region they go to. So if you look at a vessel, if you look at a vessel that goes down here by the clavicle, it's called a subclavian. If it goes into the armpit, it's called the axillary. If it goes into the brachial, it's called the brachial. So the naming, we've already done the naming. And so some of that I kind of chopped off in order for us to go through more material on things that I find is more important, because we only have a semester, to go through as we can. So I think we're gonna be okay with that. And then it just really talks about the structure of the blood vessel. And so if any questions come up, we can do an outside Zoom on it. That's totally fine. All right. So that's no biggie at all. So sorry about that.
Yeah, no worries. Thank you.
Oh, no, it's
But
important.
canvas is fine, right? If we follow canvas,
okay. Yeah, follow canvas. Yeah, follow canvas and forgive me.
Okay.
I know. I hope by now you don't go too anxiety crazy and understand that I'm trying to be here working with everybody and as much as you can work with me. This online stuff is all a little new still for many of us. Anyway, we go back to those white blood cells. We had those granule ones and then the ones without granules. And we talked about these monos are sort of in different organs most of the time, and then they become different names. And so we'll talk about those names when we get to the organs. We're not going to worry about that. And then the lymphocytes. Now, that's important. You got to differentiate the word leukocyte and to the word lymphocyte. So there's two words. Leuko means white, and lympho is the B and the T cells. But then what we do is we go through those and talk about the different characteristics of what do they do, how do they help. Like the neutrophils, they destroy bacterial stuff and by enzyme release. And then whenever you have pus, you can actually, everybody goes like, oh, it's pus. And you should actually say, congratulate your body and say like, thank you for saving my life. Because it's actually neutrophils that give up their life for your health by eating stuff and then destroying themselves in the process. So that's kind of, how you kind of look at that kind of stuff. What do they do, these different cells? And I got a thing in my throat. And so it goes through all of these different ones. We don't have to do that here. You can do that on your own. Here they go. And then from there, we also discuss, different ways. If we have too many or too little, some pathology, you see this word leukocytosis versus leukopenia. So if you're like all of a sudden you have an infection, you're going to spike up your white blood cells. You're going to have a condition called leukocytosis in reality. And that indicates an infection if you take a blood sample. Leukopenia is more of a bigger problem generally, unless, of course, the white blood cell stays up and doesn't go back down. But not enough white blood cell, that's for sure is often a problem. And that could be certain drugs, like corticosteroids could do that. So if you're a long-term corticosteroid user, you want to be, or you probably are aware of that, your immune system might be a little bit lower because of that. And so that's what that talks about. And then the next slide, also what's interesting, it talks about how do they move around in the body. And white blood cells are really cool because they get attracted by chemical signals, and then they move towards chemical signals, and they can move through the body by, they call that amoeboid motion here, which they do via the amoeboid means movement, so they can move and that way, like worms, or no, more like rolling kind of things, I think. We should visualize that. That would be really cool. But then they also can squeeze through walls, like they can squeeze through the capillary. So the capillary or the blood vessel can bring your neutrophil close to the problem where you make a cut in your skin, and then it can squeeze through the blood vessel wall and move towards the cut because it smells, chemically signaled that chemokine, they call that. And look at the process. It's really called chemotaxis. It moves towards the chemical release thing. And taxi, you take a taxi. Then soon they're going to call that chemo Uber, But anyway, that's kind of cool stuff. You guys use a little visualization on that stuff. And then after we finish with that, we're going to talk about the third type of cellular component in the blood, and that's the platelets. And that's blood little fragments. And that's, you thinking that as little, I don't know, maybe sugar no, not sugar, don't go there, but sticky things, like little sticky things that you can patch a wall with. And so you use those together with the fiber that we talked about. And when you have a cut in the blood vessel, you're going to be able to make a sticky patch with that together. And that's called coagulation, that whole process. But we're not really going too steep into this process because it's actually it's good. It's very complicated, but it's very complicated. But we don't want it to be not complicated because it really has to be sure that it doesn't happen in the wrong time. And so we're not really going into it, but just understand what you want to understand when you read this stuff is fibrinogen goes into fibrin, and fibrin is actively making the blood clot. And that word GEN, you also can learn that from that. That's a good one to know, the GEN thing. Yes, question?
Well, what's really, just a comment, what's really cool for me is I'm taking pharmacology, and we're just doing medication of the cardiovascular system and a whole chapter on anticoagulants and like all these. So it's nice the two are overlapping.
Yeah, it's interesting stuff. I mean, it's definitely very, very, it's very delicate. That's the thing. It's a very delicate process. We really have to be fine-tuning that, especially as we get older, of course. That's the thing, right? But again, that's one place where it's drink your 2 quarts of water a day will help with all of that too, because blood is a lot of liquid. It's a lot of the water stuff. So I do have somewhere, I should put that in here. There's a visual of a dehydrated blood sample and a properly hydrated blood sample, and it's not really looking good for the dehydrated one. It's kind of interesting. But anyway, yeah, this is very, very cool stuff. And then from there, the chapter moves into the blood plasma, and that's everything else other than the cellular stuff. And so that's, when you look at this, what you do is you put a blood sample in a centrifugal, in a thing that spins around, pushes the heavy stuff to the bottom and the less heavier to the top. And so the stuff that the cells with the iron in it, that's the heavy stuff. That's the red blood cells. Look at that. You got a lot of red blood cells. And then this white thing, that's the white blood cells. That's called a buffy coat. And then everything else is sort of water. And in the water, you got protein and some nutrients and hormones and stuff like that, the small components. And then that's what a blood plasma column is. So look at that, 90% is water, and then you have these substances in there. So we'll talk a little about these substances. The most interesting one about them, I think, for us to learn is the albumin, because the cool thing about albumin is if you think about you're pumping the blood, all the blood into the tissues, and then tissues need to perfuse with blood in order to get the oxygen to the cells and the nutrients and all that stuff. But the albumin stays behind in the blood vessel and doesn't get perfused, doesn't leak out of the blood vessel into the tissues. And it's a fairly large molecule. And then at the end of the perfusion, when we need to call all the champions back and say like, hey, we need to go back to the lungs to get more oxygen pickup, then the albumin is sort of a pull, a magnet that brings the liquidy and all the other stuff back into the blood vessel. The lymphatics also help. That's the double system that we sort of talk in the blood vessel chapter a little bit in. Not too much, but a little bit. But the albumin helps is that osmotic pressure. That's the pulling force that brings the liquid back into the blood vessel. So that's kind of interesting. Because have you ever wondered why isn't every, in all the tissues, there's little hearts pumping the blood back in? It's like, of course, I haven't wondered that. But when it became in like, yeah, we got to figure out how to bring that blood back in, then there's your answer. Albumin is that. So that's kind of cool to know that. And then from there, we also talked briefly about the HDLs and the LDLs, the lipoproteins, which is what carries lipid into the blood around the body. And the skinny of that is that LDL is called bad cholesterol because it takes the fat from the liver and distributes it in the body. And the HDL is the angel one because it brings the fat from the body back to the liver to process the fat. And so it sort of cleans out the body from fat. And so that's why you want more HDL, or it's why it's called good, and the other one's called the bad cholesterol. So you got to be careful, with eggs, because when they figured out that eggs were bad for you, apparently we didn't differentiate between bad and good cholesterol, HDL, LDL yet. We just called it bad altogether. And so that's why eggs are actually now really having a resurgence of important food. And I'm a pretty okay advocate of that. Not overboard, of course. What else is there to be said about the HDL and the LDL? I think that's pretty much the main part there. And then what's the rest of this chapter? Oh, yeah, then we have a brief discussion about antibodies. You heard about antibodies. We got all this virus stuff, and every virus has a little shape to it on certain parts of the virus, and then you have antibodies that are created by the body who have a shape that looks like it or fits into it. No, it doesn't look like it. It fits into that, and then it becomes an immune. On the other side, it has immune capabilities, like it can clump each other together with other things, or tag it so other cells can come and destroy that viral cell or so. And so that's where we then have this antibody. That's the little thing with the tag on it. And the antigen, which is the virus part that makes the shape that is recognizable, that's called an antigen, a disease creator, so to speak. It's not necessarily the bad part of the virus, but it's the part that we recognize. And so the body responds to it. So for us, we then get sick, right? We're going to get the sniffles and all of that. So in that respect, the antigen is the disease creator for us. And so here we have these different types of antibodies listed that are basically doing that, but all of their different, different types have a little bit different quality to it. So like one is, I forget what they really are. Some of them are in the tears, in saliva, in the GI tract. Some of them are around in body fluids, etc., etc. Some go through the mother's milk, so they passively give the immunity over to the baby. And so these are all the different groups. You don't really need to know all the, different flavors, but it's, as a concept, it's, I want you to know what they generally do, so you understand that when you further go into it. Or you can have, the conversation becomes more interesting when we talk about viruses. And then we're going to go to transport the oxygen around the body. We do that. That's pretty self-explanatory. And then we have a discussion on blood groups. And so we'll probably have you guys do that. And then we'll have that discussion maybe next week if there are questions arising from it. What's important to me is when we call a blood group, well, let's talk briefly back up. Blood groups we need because when you do blood transfusions, some bloods don't mix and match, and you got like problematic reactions on the recipient's end if you give them wrong blood. And those complications mean they die, and that's kind of a problem. And so we have to figure out which blood can you give which person. So we figured out a few markers, a few antigens markers. They make the other person sick. They're not really a virus or a bacteria, but they're still markers that have a reaction in the recipient. And so therefore the recipient's got to have some antibodies to it. And so you got this antigen antibody connection in certain people. And so when you have certain blood cells, red blood cells, they have markers on them depending on who carries them. And some people have the blood cells, they have markers on them, the antigens, and they call them A. And those people, they're fine with that because those people don't have any antibodies that attack that A thing. But the problem is if you give that blood to a person that doesn't, or that has that antibody, and then it attacks it. All right. And so what we realized then, the person that has the cells with those antigens on it, they actually generally have antibodies for the opposite, for a different marker. And we're going to call that marker B. So they just figured that out over time and they called it A and B. And so then, you can't cross them because otherwise they give a hemolytic reaction. And so that's kind of interesting. And so I want you to read through that. But the thing, and learn a little bit from it. But it's a kind of a little bit confusing. So you got to be careful and not go too fast. And so what I had to learn is the fact that when we call a group A blood, you probably know, are you A, B, or B, or negative group O, which is none of the two. You probably know that, or you can find out of what you are. And that means if you're group A, that means your blood has the antigen A on it. All right. And then you have to do step 2 from there and say, like, therefore, that person's blood, that blood A, will have antigens B in its blood. And so that way you don't confuse, or at least I don't, maybe it's easy for you, but for me it's confusing. That way I don't confuse that antibody B, is that group B or is that group A? No, it's the group A stands for, or the group B too, but it stands for the antigen. It represents what antigen the red blood cells have on them. Does that make sense?
Mm-hmm.
Good. Because, well, to me, I hope maybe you guys are good with that. To me, that's confusing. And then we have 2 more groups. We have one group that person is kind of has both antigens on them, so they are called AB. That person doesn't have any antibodies in their blood, so that's actually cool. They can actually get anybody's blood, and it's all fine. They don't have a reaction to it. But then there's the other one that has none of the markers, none of the antigens, and those have that blood has antibodies to both bloods, and that's, of course, the problem is then they can't get anybody's except their own people, their own group O blood given to them. And so that's sort of how that works when we look at the groups. I want you to work through that a little bit in your head and in a couple of questions. That way you know about it a little more, be familiar. And then we got the RH factor. And that's when you say, oh, I'm A positive. So the RH is from rhesus monkey. That's why they call it RH at the end. And it's about pigs, not monkeys. And it's, can you a pig? I thought rhesus monkey. Oh, God. Anyway. Well, the RH stands for rhesus. And it's a positive or negative system. So we don't really call it RH. We just call it positive or negative. And that's an interesting system there because it is more of an exposure. So the first exposure is a priming exposure. It doesn't make the person sick yet, but it creates the body starts creating antibodies at that point, and the second exposure is the problematic exposure. And so that creates issues when we have pregnancies, multiple pregnancies, the moment the moment the baby's blood don't match up, and then you start having interactions in a secondary exposure if you have a negative mom and a positive child. But we have ROGAM for that, but it's good to know about that. That's kind of an interesting. Look at that final footsie. I like that picture. But that's an interesting discussion there, because that's exactly the concept of when we now go into the immune system, briefly, because we haven't done much time, and talk about how the immune system functions and works. Because if we go in because that's where the exposure comes in. That's where the vaccines come in. We actually do vaccines. What we do is we want to expose the body to a virus that is not making us sick. They call it an attenuated virus. It doesn't make us sick, but it primes the immune system, and it tells our immune system what to do if that virus actually shows up, and we do get it. And so that's that primary and secondary exposure, that in the blood, in the RH, in the rhesus system. We just explained it in there, and that's exactly how it works in the body with the primary and secondary exposure when it comes to the specific immunity, like the T cells and the B cell immunity, but that the ones we take shots for and all of that. So let me actually go into that real quick. You guys have a couple more breaths left for me on that, or no? Well,
yeah.
Yeah. Good. Because the immune system, you got to take a little time and go through that stuff a little bit slowly, because it gets a little bit complicated. And then also I'm not going to be at full harping on it. I'm going to go with the basics and look in the test review back there, what's really at the end, what you really need to understand, because it gets complicated, and we want to go as good and deep as we can, but try to stay understandable in the whole thing. And so there might be some question marks that we might not get answered, but we can always have a talks offline if you guys want to dive deeper, and I can try to explain it more and do a better job that way. But the interesting thing about the immune system that I think is that this picture up here, the primary the immune system has two mechanisms, two inside mechanisms to it, a specific and a non-specific. But before that, what we have is the skin, the mucous membranes, the saliva, all that stuff that can trap bacteria that don't let the bacteria into our system at all. And so that's where that is interesting and important to understand. But then once the bacteria or the problem is in the system, then the body generally fights it initially without specificity, just generally, like all the neutrophils cut in the finger or so forth. And then after that, we have more specific fighting abilities. That's where we're like, oh, look, we've seen this virus before. And that's where that primary exposure, secondary exposure comes from. And then we've seen this virus before. So we actually are prepared for it. And we already have immune bodies, antibodies for it. And this is what the antibody count shows here in a primary exposure, secondary exposure. So that's when you get into the specific immunity. And so that's talking a little bit about that when we get to that. But the non-specific immunity, the first line of sort of defense when we get into it, that's where we get the inflammatory response showing up a lot, where we get some heat going on. You visualize you do a cut, you step on a tag or a piece of Lego or something, and you get some redness, some tenderness, some swelling, some pain. And these are all the signs of inflammation, the cardinal signs that you get, because these are the things that are happening to create these problems for us. But that's really the body saying like, hey, there's a problem, we got to fight it. So you got to keep that in mind when you just chalk down anti-inflammatories, because some inflammatory response, that's a natural process for the body. So that's one question that I think is important to ponder especially for some of us who take long-term ibuprofen, for example, non-steroidal anti-inflammatories, because it does hinder some of the repair process over time. So that's sort of interesting there. But that's a non-specific way of the body fighting what comes at it. And then from there, and then from there, I'm looking for these specific cells. And then from there, we have some specific cells that recognize, they eat this one. They eat the stuff that makes us sick, which is the antigen again, but it could be the antigen on a virus or a bacteria or whatever we don't want in the system that makes us gives us problems. And then they realize that's a problem and they put it on top of their cell and fire off a molecular alarm and say, hey, we got a problem and this is how the problem looks like. And on top of them, they have that marker. And then they also, all of our own cells have a marker on top of all the cells that tells all the other cells, hey, this is one of us. This is one of us. So it's kind of like a flag for a nation or so. And they call that the mature histocompatibility complex. I know, great name, right? Make it more complicated. But there is this one cell that then is that in between, and we call it an antigen presenting cell, APC, or also a dendritic cell because it has spikes to it. Dendrites are spikes. Gonna learn that when we talk about the neuron in neurology. But they fire that alarm, and then from there, the T lymphocytes, the T cells, which are from those lymphocytes, when we talk about the blood, they then recognize that specific antigen because they have an antibody on them that recognizes that specificity. And then from there, they can create a much stronger attack. Like they have sharpshooters in their arsenal. They have self-guided antibody bullets that they can throw around. I mean, then you start thinking of some kind of a war movie thing, and the way that they deal with that, and that's kind of cool. And that whole chapter part goes into that, the specific immunity and how that happens. And then, of course, the problem is at the end, you got to make sure you don't go overboard. And so we have also immune cells in there that are held they're called suppressors, T suppressor cells that sort of slow down the effect of what we've created. But look at that. I mean, we got like these pictures. We got like sharpshooting killer T cells, they call them. And we also do have T helper cells. That's the one when you look at AIDS that gets infected by AIDS. That's why that's such a problem, because the helper coordinates the immune response as well. And so you go a little bit into that. And so that's kind of cool stuff, I think. Well, look at these pictures, with all the spikes. So these are the other, not the T cells then, these are the B cells. And the B cells basically create antibodies and throw them around and into the system. So they create these, little self-guided missile things or whatever we want to call them if you want to go war movie stuff a little bit. And then those missile little things, those antibodies, have all these different abilities. They can neutralize a virus or an antigen host thing by covering its outside, and so it can't do anything anymore. They can clump it together and create, an agglutination that way. They can precipitate it out. They can get a lattice going so it's easier recognizable by other cells that then can kill it, like the T killer cells, for example. And then look, or they also, the other thing that they do is really cool. They actually create what they call a complement. They create a hole in a host, like here, a hole in a host. And as soon as you disrupt the membrane, the boundary between in and outside enough, the inside is going to be destroyed because you're going to get all the stuff going back and forth, and that's not going to hold itself together. So they do that too. So it's like a huge bullet that they create. So they're pretty innovative. It's pretty cool stuff. You gotta maybe watch some YouTube. I gotta go back into YouTube and try some stuff out because some of these things they visualize. Like, I don't know if you've seen the Cell movie that I got sent around from TED Talk that visualizes cell division. That's pretty darn cool. And then from there, we go into the immunity. We talk about natural, artificial, adaptive, innate. So that's an interesting, quick discussion. You could do that on your own. And we briefly talk about lymphoid organs. And lymphoid organs is where areas where pathogens easily enter and replace immune organs there. That's like our lymph nodes, our tonsils, all that kind of stuff, the appendix. In the gut, we have Peyer's patches, thymus too, even. No, that's its own organ. The lymphoid organs are the small ones. The thymus is one here behind the breastplate, and that is the teacher for the immune system. So that the school. So in there, they learn how to recognize each other. So they learn that MHC that we just briefly touched on. And then we got the lymph nodes. We understand those probably because when they're swollen, they hurt in the neck, and then you're gonna get sick. And the doctor can palpate them. They're hard nodules. They know, yeah, you probably have an infection because that means you probably have a numerous increased amount of white blood cells in the system, which means you're probably having an infection. And so that's why they do that when you go to the doctor. They palpate you here sometimes on the neck. And then the spleen is and what the lymph nodes do, as the word lymph indicates, they clean the lymph, which the lymph, as we had in the blood vessel system, is a system that picks up excessive fluid from the tissues and brings it together and brings it to the heart, and then dumps it into the vena cava by the heart, and so it can be brought back into the system that way. But on its way up towards the heart or down, but mostly up, it goes through lymph nodes, and they filter the lymph. And that's why it's a problem with cancer. That's probably where some of you heard it from. But it's also helping clean the lymph from infections. That's the idea there. So we have a lot of white blood cells in those lymph nodes. So they're like filters. You think of them as filters. And that's pretty cool stuff. So a lot of the T cells are sitting there waiting in the galleys before somebody shows up. They can shoot down. And then the spleen is one. It monitors and filters the blood, so that's an important organ. So that's the same as a lymph node, just for the blood, basically. And then we have tonsils. These also are in the mouth, checking out and making sure we don't get bacteria coming in or bad stuff. The sooner we catch it, the better, right? The less it's in the system, the better. And so you also have a lot of that stuff in the gut, because the gut is basically the outside. The tube, the inside on the tube is not hermetically sealed tissue. Everything, inside, we have to be very careful. We don't want to have infections going there. I mean, that's how people die when you have a burst appendix, unfortunately. And so we need to keep it very sterile in there, not hermetically sealed. We need to keep it sterile. That's what I meant. And in the gut, we have a lot of that tissue that also makes sure the bacteria doesn't cross that line into our sterile environment of the body. Yeah, we're like a donut. Did you know? It's got a long tube going from here, going all the way up to the back. That's the donut hole, is our gut tube. They call them Peyer's patches. I don't know why they call them Peyer's patches. But anyway, that is basically the coverage of this two fairly large chapters. I understand. So spend a little bit of time with the quizzes, go through those questions a few times, marinate that material, have that test review next to you to know that's all I care at the end, so you don't have to be too worried about it. And it's even an open book test. So really you have to understand it. You don't have to memorize it. Even I'm an advocate for flashcards, because it's ultimately you're going to memorize it. Or at least the more you do memorize it, the longer it stays in the system, so that is definitely a good process, but you don't have to. So then the other thing we're doing this week, and I can be very brief about that because it's hopefully somewhat self-explanatory, and that will be, here's my Safari. That will be the pathology stuff. So I'm going through it. Now, this is the time when you guys make sure you can connect with each other as much as, if you can't, you reach out to me. You all got my text number. So start talking to each other in each other's group. I'm also going to go back and make sure everybody's in a group. And then it's time to do some research. So you have a title. You don't need a title. You need to know a topic, what you want to do. If you haven't done that yet, communicate with each other about that. And then if you do, then the question becomes, who wants to do which question? Who wants to do describing what is the pathology? Who wants to do how does conventional medicine treat it? And if you have more questions about that, last week's Zoomy was on that a bunch. Just go. I don't know exactly where it was, but it's in there at the end, I think, towards the end. And then the third person will like, how can you as a patient support the medical treatment with lifestyle changes or alternative interventions? And so you each get one of those questions, one a person, and you each individually gonna gather source data and go to at least 5 websites. I would start with something like WebMD or depending on the question, and go look what they write about that, and then take it from there. Even Wikipedia is a good start, but go to a few sources and get sort of an idea. What are they talking about? And then you sort of get an idea of, what you want to extract and how do you want to maybe describe it, because as you read, you see like, oh, they write it pretty well. I can kind of do that a little bit or so. Or they have some cool pictures. Look some images up. And then you gather some data, and I want you to go at least 5 sources so you have a good cross-section. Take some notes, because ultimately where you want to go is you want to make either a PowerPoint or a brochure or so. I'm actually advocating for almost like making something more tangible than a PowerPoint, making like a little online PDF or something, that a couple pages. That's good too, because then we can give it to each other, and you can have, and it's like, hey, you look, I learned about what you can do about asthma. If someone in the park says, my kid has asthma, oh God, so many drugs and blah blah. They might not know that you don't. Maybe you should do the little less milk stuff or so, a little less dairy or less. Maybe they have a gluten thing too that creates phlegm. So that's another way. Think a little bit of the applications. It doesn't have to be PowerPoint. I know everybody's just resorts back to that, but gather some of the data that ultimately you can create a 2-3 minute or talking point of your main points that you sort of extracted about one of these questions. And this first, what we do in terms of now, in the next, I think it's 2 weeks. When is this due? I think it's due on the 5th, right? Which part? Come on, Slowski. Shouldn't have gone there. All right. Yeah, the 5th. So is the other one that's coming up too. So take a couple of weeks, a little bit at a time, so the material seeps into the brain cells. That's good to do that, not all in one sitting, that kind of stuff. And then gather info together and start creating a presentation with it. So gather, or whatever we talked about, gather your data together and start sharing with me what you have. On this one, I want you to share individually with me. So that way, I want you to work in groups. So I want you to also talk to each other, because at the end, you're making a video all together, but each has its own question. So if that right now, what the group means is support. Right now, what the group means is like, oh my God, I've never done a research project. I have no idea what that means. You first ask your members in the group, can somebody help me? Then you go to me. Number 2, can you help me? And then we all together figure it out that way. That goes together for the technology too. That's also a thing, because the one of the parts about this project is I want to do it, and I keep doing it even if it's online. I want to teach you as much as I can. I'm not a great presenter maybe, but at least I have some years of experience with doing it, and some points that I can give some of you to to get maybe better at, or to get exposed to, because presentations in your college experience is just something you got to get over with and do. It's just something that it has to be done. And it's a good thing. It's a little scary. It's a little anxiety provoking, but it's a good thing to overcome, because it's actually a lot of fun. And generally people have a lot of fun doing this presentation. And so start doing that and be in connection with me. As soon as you have something, start uploading it, because you can upload over and over. We can go back and forth that way. If you want to meet on Zoom, send me a text or send me something that I communicate with me. Text is better than email, especially these days. Text is definitely better than email until I'm through grading the test. All right. Is that pretty clear, or question on that front for right now?
I think it's clear.
Good.
Yeah.
That's pretty good.
Yeah.
All right. Then lastly, this week or next 2 weeks, again, this is a 2-weeker. I want you to think about, since we've doing blood stuff, and the biggest blood problem we have in our society is blood pressure. I want us to think about blood pressure a little bit. And I want us to, it talks a little bit here about blood pressure. What is it? What's high, what's low, etc. I'm not going to have you take your blood pressure. You can if you want. That's fine. We're sort of post-COVID. You can probably get it at the store if you want to. But what I do want us to do is talk about things that we can make blood pressure better. And I have a list of things that can make blood pressure better. And the biggest thing that we probably can talk about and we'll maybe work on a little bit is the salt. We can quit smoking, all that, and etc. But we don't all have that problem, thank God, or caffeine. But we could do the stress. But we'll talk about the stress stuff when we get to the brain. So here, what we're gonna do is we're gonna talk about salt a little. You learn about salt a little. You learn about how much we should eat a day or so, and then I want you to track your salt for 5 days and do the best you can to track the salt that you take into your body. Sodium, salt, same thing, right? And you take into the body. Read it on the label. Go back to that nutrition label thing that we did. Look it up on a Google a little bit. Just be a little nerdy and taking note of how much salt you're eating for a few days. And then I want you to, 5 days I want you to track. More as good as you can. Okay, don't we'll do the best we can, because sometimes it's hard. And actually, if you generally speaking, at the end of the day, you'll notice that if you cook yourself, you're probably using less salt than if it's in the store from the store, if it's pre-bought. But that, I want you to track your home-based stuff as well as you can as well. So be a little nerdy on it. And then list your daily totals, post that up, and then I want you to think about that stuff a little bit with these questions, these prompts, and that's part 2. And then we do that for 5 days within that next 2-week period at some point. And then afterwards, we're going to have us a discussion on it and see if we learned anything from it and see. And I can do it too, so I get better at it. And that's all I have to say. Anything you guys have to finish up? I know I went a little over. Sorry about that.
I have a question, but it's on the groups.
Yeah.
And then Barakat, you can tell me if you were able to see the file that I tried to share. I tried to share a PowerPoint template with my group.
All right.
Do I need to try and share it with you as well? First of all, I don't know if it worked.
What group are
you? Did you share it on Canvas?
I did, and
what group are you?
I'm group 3.
Group 3.
All right, we can look into that.
Yeah.
Let me give me a minute to finish up. I was one more thing I wanted to say, but now I forgot. So it's probably not that important.
Well,
but if you have anything your way, send me a text. Oh, we are in the class tonight. So
you guys are. I won't be.
I know we're gonna do a heart dissection. So
I know. I really wanted to be there.
But make sure you show up, because it's gonna be a lot of fun. All right.
So I tried to go to lab through Zoom last week, and I was using this link. Did I have to call? Because I'm like, oh, nobody's letting me in. So I left.
That was me.
Oh, okay.
Yeah, that was me.
Okay. Should I try again?
Yeah, we can try again tonight. We can
try
again. But
yeah, I really want to see that heart dissection.
Tonight is easier because I'm not gonna be all making videos tonight. I'm just gonna be there.
Okay.
All right, you guys. So, but yeah, come tonight to class. It's going to be a lot of fun doing a heart dissection. It's a lot of, cotton and stuff like that. So it's nice to feel the tactile feel these organs a little bit. But other than that, if you have any questions, reach out, and I'm good to go for the week. All right.
Okay.
Have a good one. Bye.
Bye.